Good morning, everybody. Thank you guys who came for the lecture. I'm going to continue a discussion of the skeletal muscular system. I actually will finish up today. I believe this is where we left off last Thursday. We had talked about the importance of calcium. And just to remind you again, from the nervous system, we know it's absolutely critical for neuron talking to the next cell down. So we need calcium in order to allow for exocytosis and the release of neurotransmitters. So it's going to be critical for muscle cell communication. It also is critical for allowing for contraction to occur because it actually binds to the calcium binding sites on a troponin complex, whereby that moves out of the way and allows for myosin to bind to actin and to initiate contraction, right? So what I want to talk about now is up to this point, we've talked about, I gave you the general anatomy. I told you about the smallest contractile units, how they work, how we get contraction, relaxation. Let's translate this to the muscle organ. All right. How does the whole muscle work? All right. So if I was where you were sitting, say over 50 years ago, we actually had laboratory classes in physiology. I mean, we worked with everything from turtles to frogs to rabbits to rats, etc. One of the things that we would do back then was we would actually remove muscle groups from, say, a frog leg, and we would attach them and measure twitches. In other words, what we do is you'd have a muscle, you would attach it to a device which can measure tension, then you would have electrical stimulation, and you can induce what's called a muscle twitch. So we talked about the contraction of a sarcomere. Remember, those are components of myofibrils in the muscle fibers, and together they make a muscle. So when I stimulate that muscle group, that will mean that if I can stimulate it, I will get contraction and relaxation. That single electrical stimulus is synonymous to providing a action potential, right? So basically, there's going to be three aspects to a muscle twitch. There will be the latent period where there takes time. Okay, we've received a signal. Remember, that has to be translated to an action potential on the surface of the muscle cells. That has to translate through the T-tubule into and induce the release of calcium for the sarcoplasmic reticulum. That then has to bind, the calcium has to bind to the troponin complex, etc. So this all takes time. That's part of the latent period. At that point, calcium is saturated, the calcium binding sites of the troponin complex, and now we have contraction. All right, so that's the upward motion. And remember, what this means is the sarcomeres are getting shorter. That translates to the muscle getting shorter, and now it is providing some sort of tensile force, right? Now, the reversal of this is relaxation. Now, this requires the pumping of calcium back into the sarcoplasmic reticulum. This requires ATP, i.e. energy. This also requires time. So typically speaking, your muscle twitch, the contraction period, is shorter than the relaxation period. Right? Your contraction period does not require the expenditure of ATP. It's already been expended in order to cock those myosin heads and get them ready. It's just waiting for calcium in order to initiate contraction. So it occurs relatively rapidly. The relaxation period can be rather delayed. Okay, now the graph about in the middle there just shows that different muscle organs, different muscle groups can exhibit different muscle twitches, right? I want to focus on this lower panel here. I want to spend a little time on this because this is absolutely important. So to put this in context before I begin, remember when we were talking about the nervous system, we were talking about action potentials. Remember that action potentials have fixed magnitudes. The only way we can discriminate in signal strength is by the frequency of these action potentials, right? Every time an action potential gets to the end of an axon, there is the release of neurotransmitters. They're rapidly degraded, so in order to continue signaling, I need to have repeated signals. More action potentials per unit time means more release of neurotransmitters. All right. Now, in the case of an action potential, that frequency will increase, but it has a limit because action potentials, by definition, are discrete wavelets. They have a fixed amplitude and they are wavelets. That means they have an up and they have a down. You can shrink that up and down, but you can never get rid of it because it represents the flow of ions across the membrane. So there always has to be, you have to reestablish some aspect of membrane potential before you can initiate another action potential. So they're always discrete wavelets. Therefore, they can never be completely fused. This does not apply to a muscle twitch, right? Remember last Thursday when I was talking about contraction, as long as the calcium is in the cytoplasm, as long as that calcium is binding to the binding sites on the troponin complex, there will be contraction. Do not get relaxation until you pump the calcium back. Okay, remember I talked about rigor mortis as well to try to reemphasize that concept. Now, so let's apply that here. We have our muscle group and we're going to stimulate it. All right, so we're going to give a single electrical signal stimuli. We're going to get a single muscle twitch, right? Now what I want to do is I want to increase the frequency of stimulation here. And what that means is I'm going up with my contraction. I'm starting to come down before I go all the way back down to a relaxed state. I stimulate another contraction. So it goes up. And then I can come down here and it goes back up. So I'm starting to get what we call wave summation, right? So I'm compressing those, contraction and relaxation to the point where they're overlapping. So that's the summation part. Now, I can continue to increase the frequency, and we see the same thing here. It starts to go up, comes down, very similar to action potentials, right? But this is as far as it gets for action potentials because they have to be discrete wavelets. That is not the case for muscle twitches, okay? So if I continue to increase the frequency, what that translates to is I am flooding those actual synapses with neurotransmitters, and it overcomes the degradation capacity of the enzymes that are in that, so they keep signaling. And that means calcium stays in the cytoplasm. It stays bound to the binding sites of the troponin. We stay in a constant state of contraction. Note this is referred to as an unfused tetanus. This is referred to as fused tetanus because we do no longer see these little wavelets of contraction relaxation because we're in a constant state of contraction. All right. So, yes, in the case of, and what we can see in this particular case is we maintain a contraction. All right. So, basically, an example of that translated to my arm. Okay. So, think about this as I'm going to stimulate a single contraction. My arm lifts up, then it drops back down, right? If I want to continue to stimulate my arm to stay up, I have to move into this area where I'm constantly stimulating so my arm stays up. In other words, the muscle groups appropriate for this are staying in a contracted state, right? Everybody with me on that? I expect you to understand and appreciate the distinct difference between an action potential, which is exclusively within nervous system communication, and then a muscle twitch. Okay, so it's completely different. And I hope that if you then go back and look at the rigor mortis slide too, because that other translates. So as long as that calcium is there, it's going to stay contracted. Okay. In the case of rigor mortis, you're dead. That's different. And this is obviously alive, but it all depends on the frequency. Those are those action potentials that I send there. Okay. Now, a couple of things. This is referred to as a load velocity curve. All right. I think I've emphasized through the introductory lectures and all about how here, and I use the example of my biceps. I'm going to induce contraction of my biceps. If I can induce a sufficient contractile force to overcome the load resistance of my forearm, what happens? This muscle physically shortens, which means it's attached up here, it's attached down here. If I contract it, give it enough contractile force, it's going to shorten, thereby lifting my forearm. I've overcome the load resistance of my forearm. All right? So let's translate that here. There is a shortening velocity, the rate at which I can shorten that muscle group. Obviously, the less load resistance, which means I can have an increased rate of contraction. Give you an example. No load in my hand, and I go up. Okay, I can go pretty fast, right? Now, if we start adding things, I don't have a whole lot of stuff here. Let's see. Here's this. It's really not appreciable, but it's slowing me down a little bit. What about this? This has a little bit of weight. So I can go like this, but I can't go like that with this because I have increased load resistance. Okay, so what happens as I increase the load, I'm going to decrease the velocity at which I can shorten. Makes sense, right? It's not horribly complicated. Right? You can practice this. I mean, when you exercise, you can enforce that concept. Now, not only as we increase rate, we decrease the rate of contraction. What we also do is we also decrease the distance of contraction. All right? So we're not talking about rate. We're just, what is the extent at which I can shorten this muscle? All right? So nothing here except this. Now I have maximally shortened this biceps. Okay? Now, if I'm taking this, I can do this, but maybe I can't shorten it. I can't bring this all the way up. Increase weight, doing that. How about this here? I can bring this up a little bit, but I cannot shorten this. So as I increase load, two things happen. As I increase load, I decrease the rate of contraction. I also decrease the extent of contraction or the length of contraction, right? Now, that gets us to the concept of isotonic and isometric exercise. Let's all go back to elementary PE classes, all right? You go to PE classes and you would do isotonic and isometric exercise. They may even use their terminology, but what is the difference? It's basically, the difference is the ability to overcome load resistance, right? So let's start with isotonic exercise. We have our same muscle group. It's attached. In this case, it's the one, instead of being attached to a load meter, it's attached to a load resistance. In this particular case, it weighs 3 kilograms. Now, I am going to stimulate contraction of that muscle. That muscle generates 3 kilograms of contractile force. This is equivalent to the load resistance. What results? We shorten the muscle, pick up the weight, right? So what you see over here is we can see that we're generating tensile force of 3 kilograms, which is enough to overcome load resistance, and the muscle shortens physically, and we lift the weight. That's isotonic, which means that we can get a physical shortening of the weight. Now, in isometric, let's further increase the load. Let's increase that load from 3 kilograms to 6 kilograms. Now we're going to induce contraction. we're going to stimulate 3 kilograms. That's not enough. We're going to up it. Our maximal contractile force is 5 kilograms force. That's more than 3 kilograms force, but it is not enough to overcome load resistance of 6 kilograms, which means that muscle does not shorten. We do not pick up the weight. However, that isometric exercise, we have produced more work, i.e., we've induced greater contractile force, but we did not overcome the load resistance. All right? What does that mean? Okay? All of you have done exercises with like barbells or something like that, and you're going like this. This is isotonic exercise because I'm contracting my muscle. All right? And I can say the load resistance was the weight of my forearm plus whatever that weight is. Isometric exercise, you actually can be expending greater force, i.e., I want to push through this wall. I can sit here all day long and I can put as much force as I can put there. I put a considerable more force than picking up a 5-pound weight, but I can never move that because the resistance exceeds my contractile capacity of my muscles. All right? So that is the difference between isotonic and isometric exercise. Yes?
How does making your muscle bigger or how does that increase how can you lift?
How does weight training increase your contractile force of your muscles? So let's talk about that. So you go to the gym and you start using like barbells and you're doing this. All right. What you're doing as you increase the load, you're increasing the load resistance. Okay. What the muscle does, the muscle adapts to the increased load that's being applied. What does that mean? That means that you're going to get more muscle cells, you're going to get bigger muscle cells. What does that mean? Overall, more contractile elements, that is myofibrils. So that's increasing your contractile capacity. So when you're exercising with weightlifting, what happens? Your biceps gets bigger because that muscle's getting bigger. And it gets there two ways. You either get more muscle cells or more fusion to get bigger. It's actually a combination of both. All right. And we're not going to discuss a Wolff's Law here. We'll discuss that when we get to bones, but we will translate it back. But when you work your muscles, they will adapt to accommodate the increased work that they're expected to perform. That's why when you are bodybuilding, you're doing exercises that are going to where your body will respond and your muscles will get bigger, right? So that's basically how that works, all right? So we're okay on isotonic isometric exercise or contraction, right? Now, the next thing we want to talk about, it's obvious that you need a lot of energy to work your muscles. You need energy in order to have power strokes. You need energy in order to pump that calcium back to sarcoplasm. You need a lot of energy in order to use your muscles, right? So where is that energy coming from? Three basic sources. The first source is creatinine phosphate that exists within your muscle cells. That's a direct conversion of creatinine phosphate to creatinine with one ATP produced. There's only so much room inside that muscle cell for different kinds of molecules, so there's a limited supply of creatinine phosphate, and it's rapidly degraded. The next level is glucose. Glucose is converted to basically pyruvic acid, and with the generation of 2 ATPs. Now, everything depends on whether there's oxygen or not. If there's no oxygen, those 2 pyruvic acid become 2 lactic acid. And those 2 lactic acid have to leave the muscle, go to the liver where they can be translated back to glucose. And I'll get to that later. If, on the other hand, there is oxygen, we have oxidative phosphorylation. You may have had it in your high school biology class. If you had one, it would be called oxidative phosphorylation, TCA cycle, Krebs cycle. All right. You could take your pick. I was taught over the decades, all three. They all mean the same thing. They all occur within the mitochondria, which are the powerhouses, the power generators of the cell. So the beauty of oxidative phosphorylation is that you can use a variety of precursor molecules to generate ATP, the most efficient mechanism within us. So in the case of glucose, we get glucose to pyruvic acid, 2 molecules of pyruvic acid. If there's oxygen, it goes to the mitochondria, it goes into the TCA Krebs cycle, and we generate 36 ATPs. 36 ATPs. Not only do we need, not just pyruvic acid, we can use fatty acids, amino acids, a variety of precursor energy sources can feed into oxidative phosphorylation, resulting in 36 amino acids per molecule of precursor. Extremely efficient form of making energy. All right. So these are the 3 types of, they're all resulting in ATP, because remember, that's the energy currency within your body. It's just that we have different precursors. And the primary difference is whether you have oxygen or not. If you have oxygen, you can utilize the oxidative phosphorylation, get efficient power generation. If you don't, you have limited capacity. The maximal capacity, energy capacity for non-oxygen is 3 molecules. 1 creatinine phosphate gives 1 ATP, 1 glucose gives 2 ATPs. Then we go to oxidative phosphorylation, 1 precursor gives 36. Yes? We'll get there in a minute. I'll get there in just about the next slide. Thanks. That'll be that is my precursor for moving into the next slide. All right. That gets us to basically anaerobic threshold. All right. So how do I run out of oxygen? Let's talk about that. But while we're doing that, let's talk about 2 types of exercises or types of physical activity. There's 2 types of, there's a type of physical activity which is centered on endurance, right? So like running a marathon race. When you run a marathon race, you're not running at your fastest capacity, because what you want to do is have this over a longest possible time possible, right? So you're not doing a physical activity at its maximal rate or extent, but you're backing off to some extent, and that allows you to go out maybe instead of seconds to minutes to hours. Now, what this translates to is you're staying below your anaerobic threshold, right? So if you're performing a physical activity that is like, for instance, marathon running, cross-country skiing, long-distance swimming, etc., etc. These are activities that are intended to take a long time. That means they're designed to stay below your anaerobic threshold. What this means is you maximize the amount of energy being generated by oxidative phosphorylation. You still are using creatinine phosphate, you're still using glucose, but in the case of glucose, you go from pyruvic acid to the oxidative phosphorylation, you use fatty acids, etc. You're maximizing, and you have a much greater selective store of sources of ATP going through this oxidative phosphorylation, capable of providing hours of productivity. Okay, so if you look up here, the little green, that is the creatinine phosphate and glucose. That's gone pretty quickly. And this dark blue here is the extent of which we can maintain our exercise output. We're staying below oxidative phosphorylation, our anaerobic threshold, which means we're maximizing aerobic oxygen-based or TCA cycles, whatever. Now, and that gets us out hours. Eventually, we'll use up those sources of precursor energy, fatty acids, etc., and gradually slow down. Because, I mean, you can't set yourself out to run a marathon and run it for 5 days straight. You're going to run out of energy sources, right? You can stay under your anaerobic threshold, but eventually you will run out of energy sources, right? But you have been able to perform over hours because you stayed under your anaerobic threshold. Now, what happens if you exceed the anaerobic threshold? What that means is, yes, you will utilize creatinine phosphate. Yes, you will understand glycolysis, but those are limited. Because you've exceeded the anaerobic threshold, that means you can't get enough oxygen to those exercising tissues in order to utilize your oxidative phosphorylation, which means you can't convert that pyruvic acid into ATP. It goes to lactic acid in the muscle cells. You cannot utilize fatty acids, etc., as alternative sources of energy, which means that you will have a little bit there, but gradually you run out of oxygen because you're overworking, you're exceeding the capacity to deliver oxygen to those tissues in order to maintain oxidative phosphorylation, and you run out of energy very quickly. Note the scale. Up here, we're talking hours, down here at minutes. This translates to up top here, that's a marathon runner who's running for hours. Okay? Here is a sprinter who's running for seconds, maximum minutes. Okay? So if you were to run as fast as you could, you're going to run out of energy pretty quickly because you've exceeded your anaerobic threshold. I don't care how much you want to run, you will not run over a few minutes before you start slowing down. All right. In the case of marathon runner, you set yourself, it's not at your maximal capacity, but it's something less than that. You're staying below your anaerobic threshold. You can keep that rate up for upwards of hours. All right. So everybody's with me on this. This is anaerobic threshold. So it's all about getting enough oxygen in order to maintain that. Now, eventually you will run out of energy sources, but this takes a long time, right? Now, I mentioned that in the absence of oxygen, we have glucose going to pyruvic acid going to lactic acid. Lactic acid then has to be, in order to recycle it, so to speak, it has to enter the bloodstream, go to the liver, where via the Cori cycle, it can be transformed back into glucose molecules. However, this is an expensive expenditure of energy because it takes 6 ATPs to make a couple of glucose molecules. Okay? So another reason to, so when you're, when you're running in place, okay, you're not running full tilt, but you're running in place, or doing some maximal level of exercise, what happens eventually? Your muscles burn. You feel a burning sensation. That's actually a buildup of lactic acid. All right. Now, next thing. I want to talk about 3 types of muscle fibers or muscle cells. And they are based on, their division is based on what type of energy sources are they using. Are they primarily designed for rapid but short duration, such as sprinting? Or are they designed for long-term exercise, such as marathon running? All right. So your 3 types of muscle cells. We have over here the slow oxidative. This is designed to maximize the use of ATP generated by oxidative phosphorylation. And on the other side, we have the fast glycolytic, which are designed for rapid bursts of activity, but limited bursts of activity. Then in the middle, we have our fast oxidative, which is kind of a mixture between the two. Let's focus on either end because that's what's relevant. All right. So over here, in the slow oxidative. These muscle cells are designed to acquire the bulk of their ATP via oxidative phosphorylation. So what happens is if we look over here, we have myosin ATPase activity. This means direct conversion of ATP. This is low in these cells because they're dependent upon oxidative phosphorylation. So their speed of contraction is also low because they're designed for slow and steady work. Dropping on down, resistance fatigue, because we're doing oxidative phosphorylation, they are resistant to fatigue. Next, oxidative phosphorylation capacity is obviously high because that's their primary source of energy. Dropping down, enzymes for anaerobic glycolysis, low because that's not what they're designed to do. Dropping on down, mitochondria, which is a source of oxidative phosphorylation, you're going to have a lot of mitochondria. Capillaries, they need blood to provide oxygen, so they're going to be highly vascularized to those cells. Myoglobin content, what's that? When we get to circulatory system and respiration, we'll be talking about hemoglobin, which is a protein within red blood cells which binds oxygen. Myoglobin is a similar protein, but existing in these fibers, and it binds and increases the intracellular concentration of oxygen, thereby providing for oxidative phosphorylation. Color of the fiber, because of this myoglobin, the fibers have a red color, right? And glycogen content is low, again, because we're focused on oxidative phosphorylation, and the intensity of contraction is also low. So these are the slow oxidative fibers. Now let's go over here to the fast glycolytic fibers. Excuse me, they're designed for rapid but limited time response, right? So their myosin ATPase is very high. Their speed of contraction is high because that's what they're designed for, but the resistance fatigue is very low. And because they rapidly exceed oxidative phosphorylation capacity. Their oxidative phosphorylation is low. Anaerobic glycolysis is high because that's their primary source of energy. Mitochondria are low. Capillaries, low. Myoglobin content, low. Because there's no myoglobin, those fibers tend to be white, right? And glycogen content is high. Intensity of contraction, high. So we contrast the two. All right, let me give you an example, right? We have Thanksgiving coming up in about a month or so. I don't care if you're vegetarians or not, that's irrelevant. All right. So let's just think about, so when you have that turkey or maybe chicken, think about, you say, I want light meat or dark meat. What are we talking about? Okay, we're talking about breast meat, which is white meat. And whether I'm talking turkey or chicken, let's just focus on the turkey. That means that those fibers are primary fast glycolytic. Let's translate that into functionality. A turkey does not fly for a living. He does fly, not the domestic ones. No, I'm talking about the wild turkeys around here. They can fly. They're capable of rapid bursts of flying because they fly for two reasons: to get away from a predator or fly up to roost in a tree for overnight, just be protected from them. So simple bursts. If you've ever watched a wild turkey fly, they primarily glide. They do a couple of quick strokes to get up and then they glide. And they can only do that for short distances. Okay? And their primary fibers are those fast glycolytic. Now, what about the legs? Turkeys walk for a living. They have to walk all bloody day. So their muscles are designed for long-term capacity, and those are the slow oxidative. Okay? The meat on the leg is dark because of this myoglobin content. And that translates into everything you see here is applied to the leg. Everything you see here is applied to the breast. Right? Short bursts of energy, long activities, right? Now, let's deal with a Canadian goose. A Canadian goose is dark mink all around because a goose flies for a living. Well, they don't all fly anymore, but most of them generally fly south for the winter and back up in the spring. Thousands of miles, long-term distance. So that's why their breast meat is dark meat. The legs, they also waddle and swim for a living, so they use those legs as well, and so they're dark as well, okay? What about humans? Okay, we don't have technically white and dark, and it's not relevant here. We're not cannibals. So let's think in terms of, let's translate this to Olympic sports, all right? Let me ask you a question. Is a Olympic grade marathon runner born or made? Made. Why?
Practice.
Why?
You have to work with tools.
Why? Why are they working those muscles?
To make them stronger so they can perform.
Well, that's stronger, but what about up here? Or when I was talking about staying below your anaerobic threshold. So what they're training for is to maximize whatever their sport is. Now you can train for to be a sprinter, but that's not the same training as you would do for a marathon runner. Long distance, short distance, okay? So you're correct. I heard 3 people say both made, and you're all right. All right. So when that egg and sperm mix, you have a preset ratio of these fibers. So all of you have different ratios of these fibers. That means that if you get mostly these, you have the innate capacity to be a marathon runner. If you have these, you have the innate capacity to be a sprinter. However, that is just the beginning. Now, by exercising and competing relevant to those muscle groups, you enhance the ability to perform. So in the case of an Olympic, I mean, in high school, it may be good enough, you know, whatever the luck of the draw gave you, okay? But if you want to be successful competing against a lot of good people as well, you have to do both. Yes.
Is it possible, because you said like some people are like predisposed to be short distance runners and predisposed to be long distance? Is it possible, like with enough hard work and practice, for like one predisposed to one to excel in the other and vice versa?
Yes, but yes. So for instance, let's say you have predominantly this. You can practice to be a sprinter and improve your sprinting capacity. However, you will never beat someone who has both the this is their primary ones and are training along this. So yes, you can even, I mean, any one of you can train to sprint and get better at it. You can train to marathon and get better at it. But you're not going to compete against someone who's a marathon, who has genetic propensity to be a marathon runner and appropriately trained. Okay. Yes.
So you're not able to develop any more hydrogen.
No. You can get more fibers too.
Okay. So
But-
basically, like certain training, would you be able to develop more fibers in a certain amount?
That's a nice segue into the next slide. Okay. So we'll use long distance and what's happening when you're training to do a marathon runner. What's changing about you? That's what your question. So basically, a number of things happen. Number one, because the manner at which you're training yourself, you're going to predispose yourself to have improved energy production via oxidative phosphorylation. So generally speaking, that's going to be improving. Okay. What's going to happen is that, well, this involves those existing muscle cells, their mitochondria getting bigger and more of them. Okay. So this is going to be toward maintaining oxidative phosphorylation. That means that you're training because you have more mitochondria and more capacity. that means there's going to be less lactic acid buildup because the pyruvic acid is going into oxidative phosphorylation. It's not building up. Your myoglobin content is going to increase because this is storing more oxygen within those cells. You're going to get higher concentrations of lipids within those cells because triglycerine, fatty acids feed into that oxidative phosphorylation. And your increased portion of energy derived from fat exceeds carbohydrates. I'm not going to get to metabolism, but lipids are there's more bang for the buck. So that there is more energy per pound for fat versus carbohydrates, right? So we'll have a lower rate of glycogen depletion because you don't need it because you're using oxidative phosphorylation. And you have, there's going to be capillaries increasing toward those muscles to make sure that you get oxygen delivered to those tissues. All of these things can change. Now, obviously, so in long-term running, think about long-term running. If you see a long-term runner, they're not bulky, long and lanky. It's different types of exercising as opposed to, say, a bodybuilder. Okay, bodybuilder is not going to win any marathon racing. Okay, they're just not training appropriately. Okay. So is everybody? Yes.
You said that everyone's born with a certain amount of fibers.
Ratios.
Yeah, certain ratio. Is it possible to early on genetically modify them to have more of these fibers?
Well, I suppose so. But I mean, in training, you're going to predispose, toward developing those different fibers. Okay. Genetic manipulation, possibly. Though I don't see that happening. There's some reasons for genetic modification that I think they're going to be worrying about various disease states first.
Thank you.
All right, now, remember, here is a biceps. It consists of bundles of muscle cells, and within each muscle cell are bundles of myofibrils. As we add a myofibril, we're increasing our contractile force capacity, correct? That translates to either more or bigger cells or whatever. So let's talk about mechanisms to increase muscular contractile force. All right? So we can increase contractile force, number one, if we have larger cells, that means more myofibrils, which means more contractile force. Kind of makes sense. Dropping over here, we have our large fibers, and then of course we obviously, another thing is we activate more of them at the same time. So for instance, when I am lifting my arm here, not every muscle cell within my biceps is contracting because I don't need them. Only enough are, and we'll get into the details later, only enough are being activated in order to overcome load resistance. If I increase the load resistance, I have to recruit more muscle cells. And that's over the far left, right? Now, tectonic contraction happens, for instance, remember that there's these elastomeric elements that attach the thick filaments to their corresponding Z discs. Think of those as kind of like a rubber band. As I stretch that, they get stretched and they want to come back. So that can increase my contractile force by utilizing that. All right. So that is over here. Now, increase contractile velocity. Obviously, to increase velocity of contraction, I need to move toward those fast glycolytic forms of fibers. And if I want to have, or a small load. And then correspondingly, if I want endurance, I then want to switch them to the slow oxidative, and then I decrease load or increase load. Okay. Are still small load. All right. So that works there. This is the same thing, but I'm just showing you all these kinds of things that can attribute to contractile force. Now, I want to focus again on recruitment of muscle cells. It all depends on what we require for that muscle to do. Now, early on, I talked about, so we can talk about something called motor units. A motor unit consists of a single stimulatory neuron and the number of muscle cells that it's innervating. Okay. It can be 1, it can be 2, 3, 4, 5, whatever. Okay. So that, and remember how it works. I don't care how many times the neural axons branch, the action potentials are proceeding down the length of those branches and everything. And that means if it's 5, all 5 of those muscle cells are getting a stimulus, neurotransmitters at the same time. All right. So I'm using a single neuron. I'm controlling multiple muscle cells, right? That's a motor unit. And these 2 examples, here's motor unit 1, and notice that it is stimulating 2 muscle cells. Motor unit 2 is 3 muscle cells. Also remember, we have, not only do we have the axon branching going to that muscle cell, you have multiple sites of contact, i.e. each one of these little dots here is a synapse. So each one of those snaps, you're getting the release of neurotransmitter at the same time. And remember graded potentials. The more of these, that means I'm generating a greater overall graded potential because we add those. All right. So that gets us to the concept of the size principle. I've kind of alluded to that. I want to reinforce that here. What is the size principle? So the one thing I want you to remember about physiology is life is expensive. Food's hard to come by. So your body will only do enough to get by. Okay? It makes no difference. So think about it this way. I recruit as many muscle cells as I need to do a particular job. What would happen if I didn't have the size principle? That means that that means that I had to stimulate all of my muscle cells at the same time. That means that instead of being able to, I would generate enough contractile force to pick this up. Okay, let's see, that was my limit. But that's maximal contractile force. I cannot reduce that force. Do I have some here? So what that would mean, without the size principle, what that means is I only have very fast. Okay. So that means I have nothing in my hand, whoop, like that. Even though I've well exceeded the load resistance of my forearm. All right. So without the size principle, I can't have any fine movements. Without the size principle, with the size principle, I do this. I can controllably contract the speed at which I do this. Without the size principle, I can't. Okay? All right. So how does the size principle work? That means that we will engage a motor unit sufficient to get the job done. This load resistance. So if we go up here, these are 3 motor units, X, Y, and Z. If you note, X are 3 muscle cells, and they're fairly small. We move down to Y, they're 5, and they're a little bit larger. We move to Z, and they are a lot bigger, and there's more of them. So obviously, more muscle cells, more contractile force, bigger, more myofibrils, more contractile force. So what that means is you're going to start to pick up this piece of chalk. This piece of chalk maybe is fine. I can use this motor unit 1. I've generated enough contractile force. In order to pick up this bag, I've generated enough force to pick this up, but that's not enough to pick it up. I have to recruit an additional motor unit. That's motor unit Y. And that allows me to pick this up. Okay. Then what that means, if I get a higher load, I'll recruit more. But that I'm adding, it'll come back in a second. See? So basically, I will and I add on to, okay? So I've recruited this one, I add this on top of this one, I add it on top of this one. Now, obviously, we can overcome the capacity of that muscle to remove that load or contract. That gets us to isometric contraction. So the force of contraction basically increases as we recruit more muscle fibers, more muscle cells. And that gets us to the end of this lecture series. Oh, by the way, I didn't tell you this before, but I'm going to let you guys go through it because we're finishing up now. Okay. So this is the last slide. And this is kind of combining the nervous system, the muscular system, etc., but focusing on muscle resistance and so on. So think about this here. Your elbow is a fulcrum joint. Okay, so here's the fulcrum back here. And then notice here is the origin of this bicep is up here in my shoulder. The insertion site on the radius. When this shortens enough and overcomes load resistance, we can pick up that weight. All right. So I don't expect you to know the details. It's just like this is driving is home. All right. So these are your skeletal muscles. And again, I will talk about the cardiac and smooth muscles in the circulatory system. And I will talk about smooth muscles in the digestive system, and et cetera, because it's in a lot of different systems. All right. So that's it. There is obviously no quiz today. There is no homework this week. That was last week and so on. I don't believe you guys have anything due until after the break. Yeah, Thursday? Okay. Well, you have the homework due. No homework assigned. The homework I assigned last Thursday is due this Thursday. There is no assignments until you come back from break.
It's on Thursday.
I don't think there's, because the quizzes are on Tuesday. I'll double check. If there is, there is. It's not a bit, okay, the one thing I guarantee you won't have, you will not have an assignment over the break. I don't do that. All right. Okay, everybody. I'll see you Thursday. Yes.
2 questions.
Uh-huh.
First,